we talk about capping, let's look at the capping complex. So here are all the parts. So you can see this twisty thing right here, and this is linked in the PowerPoint, so you can go and look at it yourself if you want, because it helps. So you go here, and this thing, this sort of twist, that's obviously the RNA. And so you can see it's double-stranded because it's got the template strand and it's got the new strand. And we'd have to look right in the center to see which is which. I'm thinking the gold is the template strand and the blue is the new strand, but I could have that backwards. Alright, this sort of big greenish enzyme, the bottom part down here, that's going to be your NERAN, so that's your NERAN active site right in there. This is your, it's like you're looking at a catcher's mitt, kind of like that, looking at you, like that. There we go. So the active site's going to be down in here, which is where you see the new nucleotide being added right on there. And then you're going to have your other proteins sticking out. So NSP8, we've got a blue one right here with a little bend in the shaft, and we've got another NSP8 sitting over here. This is the club head, and this is the rest of the club, and it sort of tucks back there. And then we've got an NSP7, which is this pink thing sticking out at the side, and all it does is stick out. Then we've got the helicases. So here's the two Rec-A domains, 1 and 2 right there, and the two Rec-A domains right over here. And those are where the enzyme, where the helicase activity is going to take place. The one on this side, here we go, so this sort of greenish one right here, that's going to be the one that we think is the main thing that's pulling the template forward. And then this one is actually kind of tilted around a little bit sideways. The idea, and nobody's yet solved a structure where they can conclusively show this, but the idea is that this one is oriented to pull the template backwards, and so there's some kind of handoff, and you can see the part that's down here in contact with the NSP8 and with the RNA that's coming out is this zinc binding domain, and that's what you have up here. And so those are the parts that may well be sensing mismatches or engaging the helicase switch, if that's what's happening. Now the other things that are going on here, so you see underneath this thing, which is your NERAN domain, if we pull backwards, the RNA is going to split. It's going to be split by NSP7, which is the pink thing that sticks out, and the newly synthesized stuff is going to go down. Down leads you to, here's an NSP10, is this little yellow thing, and it's a necessary cofactor for exonuclease activity. Here is the exonuclease domain, and then at the bottom is the methyltransferase domain. And so this would be in position to catch the nucleotides, or I actually think there may be two of these complexes that sit kind of like that. And those would then, the NSP14 from this one would be very close to where the nucleotides are coming out from the other one. And so it may well be that that's actually what's happening. Like it can do it a little bit with one complex, but if you have multiple complexes stacked up sort of head to tail, then it's probably much more efficient at doing its job. All right, so let's put that in some kind of motion. And let's shrink it down, and let's put it over here in the corner, and let's get back on top of it, and let's move this up a little bit. There we go. All right, and let's talk capping itself. Okay, so ordinarily, capping as it happens in a eukaryotic cell takes 4 enzymes. The first enzyme is going to be a terminal phosphatase. It removes the terminal, the end phosphate from the 5' end of the RNA. And remember, having a triphosphate there is something that's going to attract attention from the cell. If this is loose in the cytoplasm, which is where our coronavirus is going to replicate, then there are proteins such as RIG1 and MDA5 that are going to recognize this and are going to set off a cascade of events. Okay. After the terminal phosphatase, then there is normally going to be a G addition. So we're going to add the G, but instead of adding it the 5' end to the 3' end of the previous one. We're going to add it 5' to 5'. And we're going to lose several phosphates. We're going to lose 2 phosphates by doing that. And so we'll keep 1 phosphate here, and we'll keep those 2 phosphates there. And that is now the cap. So that the name for that process is guanylylation, or you can say G-capping if that's horrible word is too much. GMP elation, only in the case of coronavirus, it's going to be GDP that's going to be used. But it's like the GMP that's added, and here they're showing you the substrate. It was GTP, and phosphates are going to come off. All right, anyway. Then we're going to have 2 methylation steps. Both those methylation steps are going to involve S-adenosylmethionine, which is also called SAM for short. And we're going to take the methyl group off of that, and we're going to turn it into SAH, which is without the methyl group. And so we'll put the first methyl group here on that G, and this makes it a cap of 0 structure, which is like actually a totally functional cap. The cap 1 structure, and I don't know why these are 0 and 1 instead of 1 and 2, but that's what they are, so that's how it is. Cap 1 structure is going to be when we have our cap 0 structure and we add another methyl group over here. So for coronavirus, it's NSP14 N7 methyltransferase that's going to add the methyl group to nitrogen number 7 in this ring, and it's going to be the 2' O methyltransferase that's going to 2O methylate at the 2' hydroxyl group on the actual ribose ring. And those are both NSP10 dependent enzymes, although NSP10 is controlling the exonuclease part of NSP14, not the methyltransferase part. And it is controlling the methyltransferase activity of NSP16, because that's all NSP16 does. All right. Let's see that in a little more detail, if that wasn't enough detail for you, because I know you like detail. Yeah, maybe I like detail. So here is NSP9. At the end of NSP9 is an asparagine. Asparagine single letter code is N. Yeah. So this is the asparagine molecule, the side chain hanging off. And what it's going to do is it's going to attack at one of these phosphates. And if we zoom in a little bit, here we go. There we go. And this is one of those rarer cases where it's actually going to be the nitrogen here that is attacking. And so it's going to go for phosphorus. That's going to give an electron back to one of the oxygens on this phosphate. These 2 phosphates are going to go free, and we are going to have joined the first. In this case, they show a bunch of nucleotides there. It's probably just 1 nucleotide, because it'll do it with 1 nucleotide. And so this makes sense as the starter mechanism. So imagine that's just an A. There's no UUAAA after it. And so now we will have 1 nucleotide attached to the NSP9 via a nitrogen on the N-terminus of an asparagine. So an N on the N term of the N on the N on the N term of NSP9. Yeah, a lot of N's. Yeah, and so it's a nice monophosphate linkage. Meanwhile, NSP9, in addition to being able to add 1 nucleotide, and this one may be template dependent. Like if it's sitting on that stem loop, it's probably whatever nucleotides are in the loop, it's going to add the opposite one. That's probably going to help position the nucleotide and make the decision. Then it also is going to have a little capping nucleotide here. And so this is a GDP binding pocket. And so after we have put the first nucleotide on, then we think NSP12 is going to extend that, and that's great. And then we're going to have our cap nucleotide that comes on here, and it's phosphates attacking phosphates, and they're going to push back this electron. They're going to give it back to the nitrogen, and so NSP9 is going to go free, and we've now got a cap on here. And so now we just have to methylate that cap with the NSP14 N7 methyltransferase, and we have to methylate this A on the 2' O ribose, and that's it. That's how it's done. That's capping as we currently understand it in nidoviruses. Here's the capping complex. This is from the same paper where this comes from, and this is the link if you want to go play around with this structure. Rotate it in space, yeah, whatever. Decorate your room with it. It's there for you. This is showing the single complex, and this is the one that's going to be good at putting on the methyl group. So this is going to turn your G cap into a cap 0 by adding a methyl group to it. This is the complex. So when you get two of these things together, they like to link up just like this, and those would then put the exonuclease in a better position. It's really close to the helicase. It's this thing at the end, I believe. And so that's now going to be poised for proofreading. And so the idea is that maybe at first, when there aren't many copies of the replicase, then proofreading is hard. But if you can collect together lots of copies of the replicase, then everything gets more efficient, because the exonuclease from one is able to proofread for the other one as long as they link up in the right way. And there may be other things, like one may be making primers for the other one. They may be swapping over parts during subgenomic RNA synthesis. And we know that NSP14 exonuclease is important for subgenomic RNA synthesis. We just don't know why, but it must have something to do with like going too far, making something, chewing it back, and then theoretically I guess they switch templates and then continue. Something like that. Yeah, watch this space. All right, so here is the proofreading idea. So if you put in the right nucleotide, that's fine. The helicase, which is sitting up on top here, is just going to keep pulling the RNA along, and we're going to keep adding the nucleotide that matches the blue one to the end of the red strand. And so that's sitting right here in the active site. And you can see the pink thing sticking out is NSP7. So it's right there. It's kind of closing off the active site. And if we pull this back even a little bit, it's going to be right in between the template strand in blue and the newly synthesized strand in red. And so that's how we are going to separate these things so that we can then clean up whatever the mess has been. So the idea is that if we back up, here we go, there's NSP7, it's sticking out, it's going to split these apart. And waiting right down here, if you go a little further down, it's going to get to its own NSP14. But if there's two of them hooked up like that, it'll run into an NSP14 real soon. And that potentially is the exonuclease mechanism. All right, so we have a couple of antivirals that work against the replicase. Paxlovid and is one of the main protease inhibitors, and that's just a protease inhibitor, but it stops processing. If NSP12 and 13 and 7 and 8 and 14 and 16 aren't cut loose from the proteins around them, then they don't function properly. They are either inactive or almost inactive, and they're not able to form the complex that you see up here, because like the end of this thing may be attached to the end of that thing, you know, before they are cleaved. So there's no way to make this exact structure, which is the efficient version of the machine, unless we have cleaved some parts. So that's the protease inhibitor. The other ones, molnupiravir and remdesivir, if you look at those, they are not chain terminators, because let's see, 1, 2, 3, that's a 3' and they have a hydroxyl. 1, 2, 3, 3' hydroxyl. So those are not going to stop transcription or replication directly. And we use those words differently. Replication means making a copy of the genome or full-length RNA, and transcription means making a copy of a subgenome length RNA, at least in coronaviruses, or making a messenger RNA. Yeah. So how it works is remdesivir can be added like a normal nucleotide, and it kind of looks like a normal nucleotide, as does molnupiravir. When molnupiravir comes in, it's actually going to, I believe it's called tautomerizing. I think it's this little OH at the top, this little hydroxyl that's going to bounce. It's going to either jump over to this nitrogen or stay on that one. It'll kind of switch allegiance. When it's shifted one way, it's going to be read as if it's one nucleotide. When it shifts the other way, it's going to be read as if it's a different nucleotide. And so depending on which way this thing has flipped or flopped, it is putting in a mutation, and it's also kind of unpredictable what mutation it's going to put in. It's really cool mechanism. And so you're just poisoning the virus with more mutations than it can handle. Remdesivir, though, is going to jam the replicase, and does that by being incorporated. First of all, it's got to be incorporated into the chain, and then 3 nucleotides after that. So we're going to add 1, 2, 3 nucleotides to the end of it, and then when you get to the end, then it is actually the remdesivir molecule bumps. It clashes. If you try to go to 4 nucleotides away, it would bump into part of the exit tunnel of NSP12. So this is before it even gets to NSP8 or NSP13, and it's going to bump into that. And when it does that, the idea is that it then bumps back to the -3 position, and it essentially jams the polymerase. For some reason, the helicase and exonuclease combo does not seem to be able to remove a remdesivir. And so I'm not sure if it's getting caught as it's trying to be pulled the other way as well. But there is no way out for this thing. Once it is included, it is going to wreck one replicase complex and jam the RNA in it. It's like breaking off a key in the lock. Yeah, so it's effective. It's pretty good. And it's just a nucleoside analog, so this is a thing that will work at least a little bit against a wider range of RNA viruses. Like this one was originally developed to work against Ebola virus. It just didn't work well enough in human trials that it was actually worth pursuing. But against coronavirus, seems to work better. Yeah, just lucky coincidence. So there you go. That is antivirals, replication mechanism, capping all the good stuff about coronaviruses. Thanks very much.